CRISPR is a technology that scientists use to change the DNA of living organisms. CRISPR has two parts, the enzyme that acts like molecular scissors, which is called Cas9, and the guide RNA that finds the target sequence on the DNA. The Cas9 guide RNA complex cuts the DNA. CRISPR was discovered in bacteria where it helps to fight viruses. It remembers if the virus has infected bacteria before. If it has, CRISPR finds the viral DNA and cuts it. This machinery to find and cut any DNA sequence is being used in gene editing to fight diseases today. Sickle cell disease happens when instead of having regular red blood cells that are disc-like, patients have sickled red blood cells that look like a crescent moon. The sickled cells form blood clots in small blood vessels because of their shape. Sickle cell patients also have joint pains and get lethargic easily. Hemoglobin in red blood cells carries oxygen all over the body. Sickle cell patients have a mutation of the hemoglobin gene that makes the molecules stick together and changes the red blood cell shape. Fortunately, we have fetal hemoglobin. It is only made in babies, but we still have a very little amount of it in our bodies. To cure sickle cell disease, we can enable more fetal hemoglobin in patients. Using gene editing, scientists can knock out the BCL11A gene, which blocks fetal hemoglobin production. CRISPR Therapeutics and Vertex have the first FDA-approved drug that does this. I used a mini PCR kit called Chopped to do my first gene editing experiment at home. A pre-lab activity was to cut out the kit's guide RNA and find the matching sequences to the DNA they gave us. This was not that easy. Then we mixed reagents in 4 different tubes for different conditions. The reagents were Cas9 protein, guide RNA, reaction buffer, and water. After incubating at room temperature for 10 minutes, we mixed in the DNA sample. Then we incubated it at 37 degrees Celsius for 15 minutes using mini PCR's thermocycler. Next, we inactivated our reaction by adding proteinase K. Finally, we ran the samples on a gel electrophoresis and saw that our experiment worked.